Hello student, in continuity of our this program of the genetics, now I am going to discuss some other aspects of the genetics in this episode number 3. This entire genetics can be divided into the 3 sections: pre-Mendelian genetics, Mendelian genetics, and post-Mendelian genetics. Because Mendel was not the pioneer scientist who started to work on the genetics. Before Mendel also, there were a large number of scientists did their efforts in the direction of the genetics, but they never reached up to any kind of a conclusion, so they couldn't establish any kind of laws or principles of the genetics. But there were a large number of scientists who worked on that. Afterwards Mendel, and after Mendel there is a huge series of the scientists who did work on the genetics. In brief about the pre-Mendel genetics, this is also called blending inheritance or blending genetics. Because before Mendel, it was the debate that how any progeny achieve their characters. Whatever the characters that you observe in any progeny, where from they came and in what way they express, it was the debate. And here there to support this aspect, all those followers of the pre-Mendelian genetics believed that characteristics of the progeny are mixture of the character of parents. Both kind of a parent have the different characters and their character come together and they blend together, and the blended product of the character become the character of the progeny. This is absolutely wrong, because if character of the progeny is a blending product of the both of the parents' character, then what about the gender of the progeny? Then the gender of the progeny should also be a blended form. That is just a rubbish, nothing else. It means pre-Mendelian genetics that believe that the character of the progeny is a mixture of the character of the parents is absolutely wrong. So we will not entertain it in detail. And now that's the Mendelian genetics. Before Mendel, Kallreuter, Neet, Gauss, de Graaf were the scientists who did works on the plants regarding to the genetics. De Graaf, Neet and Gauss actually worked on the pisum plant, but they were not successful. Mendel was the successful person who also did the experiment on pisum plant. Here we should know in brief about the Mendel. Mendel was born in 1822 in Czech Republic in Brno, one of the region area of the Czech Republic, and in Augustin Montessori he actually studied where there he appointed as a teacher also. Later on Bruno Montessori actually recommended him to further study, so he went actually into the Vienna University in 1851 to 1853, where there he studied about the physics, mathematics, botany, plant physiology, entomology. And afterwards, as he returned back, he started work on the pisum plant in 1856, and his experiment continued from the 56 to the 63. After this journey of the 1856 to 1863, he reached up to the some conclusions. The paper of these conclusions he actually publicly presented into the Brno Natural Sciences Society, and ultimately they were published in 1866. But it was really a biggest unfortune of that century that no one actually gave the attention to the Mendelian work. What kind of a basic principles, laws of the genetics that he established was not in notice of anyone at that time. Later on, he get engaged into some other activities of the Montessori, and he never got the time to do some more experiment or to convince the others about his findings. In 1884, because of the chronic renal failure, Mendel died, and afterwards in 1890, three different scientists, Hugo de Vries, Schermark and Karl Correns, independently did work on the Mendelian experiment, and they realized that what Mendel did in his experimental works. Later on in 1900, these results were republished, and then the world actually become aware that what Mendel actually did. Here there it is really very crucial that we should know that why the Mendelian work was not actually identified at that time when he published it in 1866. There are some reasons. First of all, the communication at that time was very poor. Second thing, the magazine that he chose for the publication of his experiments or the conclusions of the experiments, Brno Natural Science Society, was a very limited magazine. So definitely the results actually not reach up to the general people who are actually concerned with the genetics or the genetics type of studies. Third thing, that before 1866, you all are aware, the year 1859 was very crucial when the Charles Darwin's book Origin of Species was actually published, and we know that majority of the scientist fraternity was actually engaged in the study of origin of species. That is why the attention of these scientists never goes for the Mendelian work actually. One more thing I would like to say, that Mendel was actually ahead of the time. He was, the imagination, the work that he did was actually far away from the era actually, because the people were not aware about the DNA, not aware about the gene, not aware about the DNA content where there it regulate to the character, and he was giving the explanation that how DNA is regulating to the expression of the character. That was simply, you can say that he was ahead of the time. One more thing I would like to say, that in work of the Mendel, there was a huge involvement of the mathematics and statistics. That is why also biologists or those who are concerned with the study of biology actually scared about the study of these mathematical and statistical analysis. One more point I would like to share with you, that at that time when the work of the Mendel didn't receive the proper attention of the other contemporaries or the scientists or the general people belong to the science. At that time, some other scientists actually suggested Mendel to work on one more plant, and the plant was the Hieracium. When on Hieracium Mendel did experiment, same kind of a experiment that he actually already executed on the Pisum plant, the all results were deviated. All the results that Mendel found after working on the Pisum plant totally get changed when he did work on the Hieracium. It means he simply started to think like that maybe my result on the Pisum plant was just a chance, because if they are the universal laws of the genetics, then they should also be equally applicable on the Hieracium plant. But it is not the truth, because in case of Hieracium the results get changed. The reason was different, because Hieracium is such a plant where there naturally the seed or the progeny not arise by the usual mean of sexual reproduction or the usual mechanism of sexual reproduction. This is the apomictic plant where their seeds arise by the apomixis phenomena, by the agamospermy phenomena. I hope you are aware about these terms, because in the junior sections you have studied about the apomixis in the sexual reproduction in flowering plant. So because Hieracium was a apomictic plant, that means the character of the progeny was not determining by the two parents. Here the character of the progeny was actually determining by the single parent. So how could it explain to the laws of the genetics that was actually published by the Mendel? That was the reason. But anyway, after all these points that I share with you, poor communication, publication of the result in a very local or regional magazine ahead of the time, bold use of the mathematics and statistics, and Charles Darwin's book Origin of Species, Hirasian's experimental results deviation, all these things ultimately become reason behind such a avoidance of the Mendelian work. But ultimately, after the work of Karl Correns, Schermark and Hugo de Vries, the things come out, and Mendel actually regarded as the founder, as a father of the genetics, because he did a fantabulous job about the mechanism of inheritance of the characters. So here it was just a brief introduction about the Mendelian genetics. We will come back on this Mendelian genetics because it is the subject that we have to study in the coming classes. I would like to give you one brief account about the post-Mendelian genetics also. In post-Mendelian genetics, there were some scientists who remain involved. One was the Batson. You are familiar with this word genetics. The term genetics was actually introduced by the Batson. Batson is also regarded as the father of animal genetics because he did a lot of work on the genetics of the animal beings. And the term homozygous, heterozygous, filial generation were also actually proposed by the Batson. After Batson, one of the most important scientists was the Morgan. Popularly, we call him as a father of experimental genetics. He discovered so many things for the genetics: discovery of the sex linkage, discovery of the linkage, discovery of the crisscross inheritance, discovery of the gene theory. All these things were simply because of the Morgan. Means Morgan did a fantabulous job in the genetics. Means after Mendel, Morgan was the scientist who was actually the pioneer scientist who did a huge experimental work on the genetics. Afterwards, Muller was also one of the genetists. He did work on the actinobiology and cytogenetics. Actinobiology means study of the influence of the radiations on the human health, because he did a large number of experiments on the mutation by using the ultraviolet radiations, X-rays, etc., and with help of which he did a lot of work in the genetics field of the genetics. Actinobiology, that means study of the influence of the radiations on human health. He also did work on the cytogenetics, means cytology plus genetics. It means the concern is a nucleus associated studies. One more scientist after Mendel was the Garrod. He is also regarded as the father of human genetics basically, because he did a work on the genetics of the human beings basically. You all are aware about the disease alkaptonuria. Alkaptonuria was the first human genetic disorder to be discovered. That disease was actually discovered by the Garrod. He did a lot of work on the alkaptonuria, that is popularly also known as the black urine disease, and he published this book actually, or the results of this disease, in a book. That the name of that book, title of that book was the Inborn Error of the Metabolism. Inborn Error of the Metabolism was the book in which this disease alkaptonuria was actually published. So these were some of the crucial scientists which were actually involved into the genetics after the Mendel. So after study of pre-Mendel genetics, a brief introduction of the Mendelian genetics and post-Mendelian genetics, we are coming back to the Mendelian genetics. In Mendelian genetics, as you all are aware that the Mendel did a lot of work on the various kind of plants, particularly in the Pisum plant that he did work. Various term I use with respect to the Hirasium, but the pioneer work that he did is actually on the Pisum plant. Here there in the Pisum plant, what kind of experiments he did, what was the approach of his experiment, that we will discuss here. You just remember, Pisum plant was selected for the study by the Mendel have some reasons. Why study on the Pisum was such a successful one, that because of which he became capable to establish the laws of inheritance? Yes, there are some of the reasons why the selection of the pea plant was such a crucial one. In the regions of the success of Mendel, whenever anybody asks you that what is the foremost reason because of which he gets succeed, he becomes successful as compared to the other contemporaries that also did work on the Pisum, then you should say the foremost reason behind the success of the Mendel was the selection of the pea plant, selection of the pisum plant. Pisum have some features because of which it is the ideal material for the study of genetics. What are these features? First of all, short lifespan, means the pisum plant actually complete their lifespan as well as their life cycle within a couple of weeks. It means one plant generation when finished, the second generation arise immediately. It means the scientists have not to wait for a long time. Second thing, large number of progeny production, means when one plant participate into the reproduction, it will give rise to a large number of progeny. According to mathematics, as you all are aware, or maybe, that if sample size is a larger one, then the deviations of the result, the chances of the deviation of the result would be the minimum one. So for the genetics, if any plant or if any of the member that you are using as a object for the study of the genetics, then its progeny production or the number of progeny production should be bigger one. That was the feature already present into the pisum, large number of progeny production, so that the chances of the deviation result can be minimized. Third thing, the plant have a sharp contrasting characters, means as you already know about the contrasting character, tall, dwarf, they were sharp, means the tall was as tall and the dwarf was as short, then anyone can easily recognize that it is a tall, it is a dwarf. Flower colors were so sharp, violet, white, means easily we can recognize. Seed shape, round, wrinkled, can easily recognize, means sharp contrasting character was also one of the reasons that make this plant suitable for the study of genetics. One more reason, the flowers of this plant were bisexual and naturally it was a self-pollinating plant. But by man-made efforts, you can also perform cross-pollination. Means genetically there was no barrier that if you want to perform cross-pollination, then plant cannot oppose it. The cross-pollination is also possible. Naturally, it is doing the self-pollination. In this way, it is also a really very significant one. So these were some of the reasons because of which pisum become a very ideal material for the study of the genetics. So selection of the pisum plant, one of the foremost reason behind the success of Mendel, we discussed here. What was the approach actually Mendel used in his experiment, that I would like to share with you. Whenever Mendel did his experiment, he always go through the three fundamental crucial steps. Step number 1, creation of pure line parents. Whenever he did crosses in the plants, he always used to the pure line parents, because he believed that if character is in a stable form within the parent, then only we can study the pattern of inheritance into the progeny. So always he used the pure line parents. I have already discussed with you that what is the mean of pure line, a group of homozygous organisms. Second step was the crossing between pure line parents. To obtain F1 hybrids. These pure line parents that he selected allow cross pollination so that the product of this sexual reproduction, that is the first filial generation, he can obtain. That is the second crucial step. And third, selfing of F1 hybrids to obtain next generations. These were the three fundamental steps, crucial steps that Mendel always used during his experimental journey. Creation of the pure line parent, crossing between the pure line parent to obtain F1 hybrid, and then selfing of the F1 hybrids to obtain the next generations. During this experimental journey, Mendel also used two unique events or mechanisms. One was the emasculation and second was the bagging. Suppose this is one of the pea plant and this is the second pea plant. Suppose this is the tall and this is the dwarf. I already said to you that it is also producing the bisexual flower. It is also producing the bisexual flower. Out of these two plants, randomly, any one he considered as a male plant and the another one he considered as a female plant. Although remember, the flowers are bisexual, but he considered one plant as a male plant and the second plant as a female plant. Anyway, if it is our female plant, then its only stigma is desired. In case their pollen grains means anthers or the stamens are the desired. In this case, here there, because the stigma is desired, but beside the stigma, beside the pistil, the stamens are also present. And these stamens can also dust their pollen grains over this stigma, means undesired self-pollination. To prevent this undesired self-pollination, he used one approach that is called emasculation. Emasculation. When the flower is immature, or you can say when the anthers are immature, at that time, from this bisexual flower of the plant to which he considered as a female parent, he removed the immature anthers. Removal of the immature anthers, from the bisexual flower that is considered as a female parent, so that undesired self-pollination can be prevented, is known as a emasculation. Means in emasculation, there are conditions. Anthers should be immature, flower should be bisexual, anthers should be immature, flower should be bisexual, and this bisexual flower should be considered as a female parent. Then if you remove these anthers, then this process is known as a emasculation. And for what? So that undesired self-pollination can be prevented. So suppose this is our plant, this is the pistil, this is the stigma, all these anthers I will remove, then this process called emasculation. Means now this plant is safe from the undesired self-pollination, but still there is the chances of the undesired cross-pollination also. So by using butter paper, he prepared some ball-like structures, and that is called bagging. Means there are two events, emasculation to prevent what? Undesired self-pollination, and bagging to prevent what? Undesired cross-pollination, so that only the cross between the desired male plant and the desired female plant can made possible. These two events, emasculation and bagging, were essential that he used in his experimental journey. In this pisum plant, he ultimately selected, because initially he actually scrutinized the large number of characters, but out of these various characters, ultimately he selected only 7 character, contrasting character, on which he did his experiments. I would like to share these characters. Flower color, we know here that the dominant phenotype, and this is the recessive phenotype. In case of the flower color, violet color was the dominant, and colorless condition, white, recessive. The gene that controlled to the flower color was located on the chromosome number 1. associated chromosome. Associated chromosome. Second, seed color. You should be careful. In the seed of the pea plant, color is present in the cotyledons as well as color is present into the seed coat. Mendel, when used the word seed color, then he considered to the color of cotyledons, not of the coat. Careful, seed color for the Mendel means color of the cotyledon, not the color of seed coat. In this context, yellow color of the cotyledon was dominant and green color of the cotyledon was the recessive, and the gene for this character was also located on the chromosome number 1. Then, height of the plant, tall was the dominant character and dwarf was the recessive character. This gene was located on chromosome number 4. Shape of the pod, inflated pod was the dominant and constricted pod was the recessive. Gene for it was also located on the chromosome number 4. Position of the flower, axillary position was dominant and terminal position was recessive. It was also located on chromosome number 4. Pod color, green color of the pod was dominant, yellow color of the pod was recessive. It was located on the chromosome number 5. And the last, seed shape, round shape of the seed was dominant and wrinkled shape of the seed was recessive, and it was located on the chromosome number 7. So these 7 contrasting character on which Mendel did his experiment, and in what way he did experiment, that we will study. Here there I would like to say that these chromosome numbers means the concerning characters gene are located on which chromosome. This thing was not in notice of Mendel. This dissection, this genetic dissection of the character, that this character associated gene is present on which chromosome, actually came later on the Mendelian studies, because at the time of Mendel what he assumed actually was totally different. He believed that the factor that regulate to the character are present on a chromosome. It was not in his notice. He believed that the factors are present, where they are present, how they are present, that was not in his vision, that he was not aware about it. This concern, these number of the chromosome on which these genes are present, it was discovered by another scientist, Blixt, who actually did this experimental dissection of the genetical characters. So you just remember, approach of the Mendel was here, they're totally different. These 7 contrasting character, the dominant form and the recessive forms, should be in your notice because they are very crucial. In exam, anyone can ask that select out the odd one, then you should be familiar that which set of the character is a dominant one and which one is a recessive. That should be in your notice. So here there, after the selection of these 7 contrasting character, he started his experiment. Initially he started to work by considering only one gene pair, means one allelic gene pair regulating to the one character. That is called monohybrid cross, means a cross in which he considered only the inheritance of a one character, means inheritance of a one allelic gene pair. That is called monohybrid cross. And on the basis of monohybrid cross, he concludes some postulates that ultimately become the laws of inheritance. And after the successful understanding of the monohybrid cross, means study of the inheritance of a single character, he started to study simultaneous inheritance of the more than one character, means two character, that is called a dihybrid cross. So in the same sequence we will study first monohybrid cross, their postulate, and in what laws they converted, that we will discuss first, and later on we will shift towards the dihybrid cross, means the actual genetics now we are going to start. Okay, so first in this section we will discuss about the monohybrid cross. So in this section we will finish it. In the next episode we will discuss about the monohybrid cross and dihybrid cross and so on. So about the Mendelian work and in what way he did his work is now up to you now in the next episode. Thank you very much.